Okay, well, yeah, thank you for inviting me to speak here. So I have this sort of very generic title, High Throughput Sequencing Data Analysis. This is your problem, and Kun already mentioned this. So you prepare a library, and that's, of course, where all the art is, and what kind of biological question do you want to ask. It, in the end, always comes down to a library of lots of DNA fragments. You put it into this wonderful machine that Pearly is running, and then you get this output file over there, which has sequence information, and of which I show you just the first few lines. But in reality, you have a few 100 million of those. And then so what, right? And so my interest is really in developing tools and algorithms that then allow you to analyze this kind of data. And so I'm sort of mainly interested in the kind of projects that are not the standard analysis, but where some new algorithms and some new tools have to be developed. And what I will do in this presentation is I will go through 2 or maybe 3 different projects, sort of just touch them on a very superficial level, of course, to just give you an idea of the kind of things that we are doing. So the first one is this DNA methylation analysis that Pearly already introduced you to. So we developed this workflow here, how we deal with data that comes from the methylation pull down. So Pearly's group first prepares this library, which in this case is prepared by pulling down DNA with a protein that preferentially binds to methylated DNA. Then this will be sequenced, and so then we basically get more DNA from places in the genome that are more methylated. Then we do this quality control that Pearly already mentioned in order to exclude or be able to send to resequencing samples that just are not up to the quality standards. Then we have to remove duplicate reads because sometimes in the process of library generation you just get artificial multiplications of individual pieces. And then we map reads to the reference genome. Okay, so this is basically a fairly standard analysis. So we ask where is the age read in the reference genome. What we then have to do is we have to take our reads, which are only the ends of the fragments that have been pulled down, and extend them to the length of the fragments that we know about. And then we have to normalize by the number of reads that we had. Because if today's experiment gives you 100 million reads and tomorrow's gives you 200 million reads, that's just because you put a different amount of stuff into your sequencer, and you don't want, I mean, that doesn't have any biological meaning, right? So we have to take that out, and then eventually we will determine the overall methylation, and mind you, methylation in every single place of the genome. And just to show you an example of how this data then can look like, I show you here example data from a study of a drug treatment in AML, where we show methylation before treatment on the x-axis, methylation after treatment on the y-axis, and every single dot on there, and I'm sure you can't distinguish the single dots, but every single dot is one CpG island of a set of patients, and the black line here is the diagonal. So if the dots were along the black line, that means nothing changes between before and after treatment. If you have something below the line here, then that means the methylation is lower after treatment than before. So you have hypomethylation, and if you had something up here, you get hypermethylation. And so you see clearly that this drug really reduces dramatically the amount of methylation in essentially every single CpG island. And the different colors, the red and the blue ones, distinguish patients that eventually reach CR. These are the blue ones. And that don't reach CR eventually. These are the red ones. And so you see very clear differences. This is for CpG islands. This is for gene bodies, and you see in gene bodies things can look completely different. So here we are fairly closely around the diagonal. So you see on average not much changes, but still there is some genes that go down and some genes that go up. And of course those lists of genes are then available and can be analyzed afterwards. So this is sort of just to give you an idea that we really can see methylation at every single place in the genome and then mine that kind of data. So A second example is a non-human example, and so therefore maybe the least interesting for you. But I nevertheless wanted to mention it because I think it's a very cute project. And this is on RNA editing. So RNA editing happens if you go from DNA to RNA, but something happens and it's not an exact copy. And on this project, we looked at this slime mold here. And here's an example of the DNA sequence and the mRNA sequence below. And you can see these extra bases here. There's extra C's in there that are not in the DNA, and somehow the slime mold puts them in there. Nobody knows how it knows where they go, but they are always there. Sometimes it also puts other stuff in there. So here is a CU together, and there's also U insertions and C to U conversions, and we wanted to know where do they really go, right? And so we prepared an RNA-seq library. So we took all the, and this is all happening in the mitochondria, all the mitochondrial RNA from those bugs, sequenced it, mapped them back to the mitochondrial genome, allowing for insertions, which back when we did it, the regular tools wouldn't do. And then once you have them mapped, then you know where the insertions fall, and then you know what the RNA sequences really are. And then you can start looking for those genes with BLAST. We found over 1,000 editing sites. Mind you, that genome is only 60,000 bases long, so that's really a lot. We were able to quadruple the number of annotated genes. We found new types of editing, G insertions and A insertions. We also found for the first time intergenic editing. So before people said, well, this happens only within genes, but it actually also happens between genes. Lastly, a project with Patel where we looked into short tandem repeats. So a short tandem repeat, as you might know, is a genomic region where one short sequence is repeated many, many times. And typically, the number of the repeat units is variable between different alleles in the population. So here's an example of such an STR, which is called TPOX. So you have a distinctive 5' sequence, you have a distinctive 3' sequence, and then you have this AATG, which repeats in this allele 5 times, in this allele 6 times, and in this allele 7 times. And well, people are interested in this because that's what the law enforcement uses. You know, when you in CSI see, oh, we are doing a DNA sample to identify a person, they're actually counting how long these things are in each of those people. And we asked, well, can you actually reconstruct those from high-throughput sequencing data? Because maybe law enforcement may want to use high-throughput sequencing data, and maybe you, when you do a clinical study, may want to make sure that you really anonymize your patient, and you are not able to actually go to the FBI database based on the sequencing data you give us, and we can pull up who that was, right? So it turns out that the only reads that actually help you are the ones that cover the STR from beginning to the end, but it turns out that the regular mapping approaches, they just throw those reads out because they say, oh, this doesn't look like the reference if this read has a different number of repeats than the one in the reference. And what I show you here is just a summary. The red arrows are what sort of the reference data set, so the actual sort of FBI kit is telling us. And the blue bars are the read counts that we get with our specialized algorithm based on a large sequencing data set. And as you see, for the most cases, we are actually able to pull out those numbers from the high-throughput sequencing data. Okay, so with that, I would like to close and just say that there's a lot of other projects going on, and they are all in collaborations with different people. So just like Purdy said, I really live off collaborations with different researchers, and certainly would be happy to talk to anybody else about possible collaborations. In conclusions, just high-throughput sequencing is really a great way to get insight in many different biological and biomedical problems. While these are people from other projects, the actual list of collaborators on just those that I talked to you about today are these, but I guess I'll go back here. Okay.
Thank you, Ralph. And I, at this time, would like to invite the speakers to join the Bring Your Own Chair session. So drag your chair to the front. And this would be a good time to have a discussion. As you saw during the course of the, yeah, can you maybe grab your chair here and then pull it up here and grab a seat? So yeah, thanks. So there are lots of strengths in the campus, and we are continuously developing those strengths. But a key emphasis of the center, the CCTS, is to make sure that our researchers can access those strengths and can uplift the research they're doing by appropriate utilization of these technologies. We have the speakers here. If there are folks in the audience who would also like to speak about other technologies, you're welcome to do so at this time. And now the session is open for discussion. As we do that, can I ask Raghu, the type of projects that you're currently in, is this still in the exploratory phase that you're trying to connect the utility of phenotyping to health outcomes, or do you already have some real headway where you think that phenotype B of the nucleus or the organism has something to do with functional outcomes?
I think when it comes to the more...
I'll get you this so you can be on record, yeah.
Good. Equipment, okay.
Thanks, yeah.
When it comes to coarser scale resolutions like MR and so on, I think it's closer. I mean, we've had some successes there. For, again, when it comes to pathology, yes. But when it comes to more mechanistic studies, they're pretty much hit and miss. So yes, I mean, I think one could say that for certain kinds of questions and certain kinds of topics, yes, it is quite close.
Right. Yeah. Questions from the floor? Yes, Juliana?
Congratulations to Dr. Swan. You mentioned so many different public accessible libraries. So would it be possible for us to initiate a type of collaboration based on existing libraries, or it should be set to your private data?
So you mean how to use public data? How to access? Yeah.
Would we use your facility to collaborate on the, for example, getting information about from the public sources of data with your help?
Yes, this is actually analyzing public data is actually listed as part of the profiles for the biomedical information share resource. So definitely we can have a lot of discussion on those. Yeah. So we do help quite a few projects on using the public data to generate hypotheses, as you can see. So public data, I think the great thing is it can help to generate a lot of hypotheses, or can help to complement what you already have, to boost you already have. Yeah.
It's IRB massive, right?
Exactly. So it's great to help generate preliminary data, even preliminary publications. Like last year with Carmen sitting there, we actually had a paper purely using public data for pulmonary fibrosis, so, to identify certain genes for those. Yeah. Okay.
All right. As we wait for more questions, the OSU RNA Center. Does the center have core facilities, or are people expected to go through Pearly here, or do you have some part of the operations that you provide directly from the OSU RNA Center? Ralph?
Well, so the Center for RNA Biology does not have any core facilities. It's really a community of people. It's about 30-some PIs that all work in RNA biology one way or another. And I mean, it's a great resource because basically any question that you could have about RNA, you probably find an expert here at OSU. It's really one of the big strengths in the biological sciences. But I mean, so it's not a facility per se. But I mean, the website is rna.osu.edu, and if you want to contact, you can contact me certainly, or the director, who's Dan Schoenberg. If you have any question about RNA biology, there's a lot of people that are happy to collaborate and help.
And this is, I understand, a very strong facility, and if you folks have not seen that, go to the website if you have questions. Yes, yeah, go ahead. You have a question?
I have two questions. So one, this is a general question. Do you have facilities here at CCTS to do a metabolomics platform like it was on the morning? From your experience, Dr. Berger, can you do that kind of analysis using serum from patients and find all the amino acids? Or
we know we do metabolomics at some limited scale. Do you know any better?
I know there is a board. I think you can run But I forgot who runs the core. There is a core facility. You can do some something like using MassPack or using
so Kari Greenchurch.
Yeah.
Kari Greenchurch runs that, and I don't know if they have like some of the more full-blown metabolomics cores that we have in the country, but to some limited extent, so I would recommend, and she resides in this building too, so should be able to contact and find out. Yeah. Yes, please.
I have two questions for Dr. Yang. The first question is your presentation mentioned some drug can change the promoter methylation status. So is this a global change or a promoter-specific change?
Okay. Actually, I can just... Yeah. Keep going. All right. So yeah, actually, the data that Dr. Bundschuh showed is that it changes because the method that we were using, it's a capture-based method, and it requires 4 to 5 methylated CG in that stretch of DNA to capture it. So it's actually more sensitive towards, the CPG island and promoter area where there's more CG. But we do see changes whole genome-wise. Probably it's our data actually shows that in the non-promoter, non-CPG island area, we have a mix of hyper and hypomethylation that's going on. In the promoter island and the CPG island area, probably 95 to 99% of the changes are hypomethylation with these decitabine. The drug that we're actually looking at is an AML. It's decitabine given to older AML patient that has not been treated with other drugs. And we were looking at the pre-treatment information and after the first course of treatment. So even then, just with one treatment, we're able to see major changes in the patient material.
So there is no, you cannot change promoter methylation on the single CPG island, right?
We could.
You could target for certain...
No, it's a global change. It is a global change, but there is certain pattern that we observed that it targets certain genomic region more heavily than others, and we were able to show that the drug actually heavily targeted ends of a lot of chromosomes. That manuscript just got submitted, and so yeah.
All right.
The second question, you mentioned that methylation heat map. Is that a public available tool, or we need some people to help us?
So what we are providing that as a tool for our users, because the data, just as I was saying, one lane of data can be 260 million pieces of data. So it's very hard for biologists to have a handle. So we turn the data into a heat map format so that they can look at it, very intuitively. And so if it happens to be, your data or your collaborators' data, we will be able to share that with you. Otherwise, probably at this stage, it's a service we have for our user. Thank you.
Any further questions? If not, we have to get ready for the 1:30 session. I'd like to thank the speakers and thank all of you for attending this session. Session is adjourned.